DNA acts as the encyclopedia having all information for the growth of the organism. The genetic code is termed universal as it is found in all living organisms, even viruses. However, the genetic code is also referred to as degenerate, as there is more than one base triplet that codes for one amino acid. The copying of the information contained in the DNA to the mRNA is called transcription. Turning mRNA into a polypeptide chain is termed translation. In transcription, RNA nucleotides found in the nucleolus are used to make a copy of DNA with the help of the enzyme RNA polymerase. Only one of the strands of DNA is ever copied, and this is the antisense strand. Thus, mRNA produced has the same code as the sense strand. RNA polymerase then adds RNA nucleotides using complementary base pairing. Nucleoside triphosphates are used to provide the energy. Thus, the triplet sequence codon, GCT, on the DNA is translated to CGA on the mRNA, and this causes tRNA to bring on the amino acid arginine. Before the mRNA can leave the nucleus, it is edited. The introns are removed, which is the non-coding part of the mRNA, and the exons remain. This makes the mRNA much shorter, and it now only contains the codons for the amino acids required. mRNA now leaves the nucleus and enters the cytoplasm where it attaches to a ribosome. In translation, the small unit of the ribosome attached to the mRNA and moves along until it recognizes the start codon AUG. The tRNA with the complementary anticodon now brings in the amino acids methionine. The tRNA carrying methionine attaches to the P site on the smaller subunit. Initiation of protein synthesis has now occurred. Each tRNA is specific to a particular amino acid. The specific amino acids are attached to each tRNA by the tRNA activating enzyme using energy from ATP. tRNA is now used to bring in the correct amino acids in the correct sequence, using complementary base pairing. This is the elongation phase of translation. As this occurs, the ribosome moves along the mRNA, adding further amino acids. Termination occurs when a stop codon is reached. At this point, the tRNA detaches and the large subunit, small subunit, and mRNA separate. In this way, the correct amino acids are arranged in the correct sequence, resulting in the production of a particular polypeptide. Thus, one complete sequence of codons on the DNA is translated into a particular polypeptide. The sequence of codons on the DNA is referred to as a gene. To speed up the process of protein synthesis, more than one ribosome attaches to each mRNA. Thus, many polypeptides can be produced at the same time. Several ribosomes make up a polysome. The process of transcription and translation is now complete.